The human body has many surveillance mechanisms for detecting pathogens. Phagocytes are able to detect foreign cells by looking for molecules on the surface of those invaders that are not found in the human body. Adaptive defenses, such as B cells, antibodies, and cytotoxic T cells, are looking for a specific antigen on the surface of the invaders. If a microbe can hide from these surveillance methods, then it's more likely to reproduce within the body. When immune surveillance cells, such as macrophages, are looking for invaders, they search for surface molecules that are not found on host surfaces, such as peptidoglycan. Capsules that surround some bacteria are usually composed of polysaccharides similar to molecules found on host cells. Thus, when a macrophage encounters an encapsulated bacterium, it doesn't recognize it as foreign and ignores it. This is a strategy used by Streptococcus pneumoniae, a common cause of bacterial pneumonia. Even if a bacterium is engulfed by a phagocyte, it may not necessarily be killed. The tuberculosis bacterium is able to survive within a phagocyte by preventing the fusion of lysosome and phagosome. This keeps the digestive enzymes away from the bacterium. Bacteria such as Shigella and Listeria can actually escape the phagosome and live within the cytoplasm of the phagocyte. Even if the phagosome does fuse with the lysosome, some microbes are not affected by lysosomal activity. Leishmania, a protozoan, is able to resist the lysosomal enzymes. Legionella and some staphylococci inhibit the pathway that leads to oxidative killing within a phagocyte. Some bacteria, including the tuberculosis bacterium, actually reproduce within the phagocyte. Several different types of microorganisms are capable of hiding from host defenses by changing their surface antigens frequently. These changes may occur as a result of mutations or genetic recombination. RNA viruses, such as influenza virus or HIV, frequently exhibit antigenic variation on their surfaces. Trypanosoma, the organism that causes sleeping sickness, has a surface glycoprotein that undergoes frequent changes. In all of these cases, it is the survival of the fittest. The microbes with the undetectable antigens are the ones that are least likely to be detected and most capable of reproduction. One way in which pathogenic bacteria are able to create an infection and cause disease is to have certain traits called virulence factors that help them attach to or penetrate the host tissues and to escape host defenses. One group of virulence factors are enzymes secreted to help the pathogen to penetrate tissues, allowing them better access to nutrients and helping them to reproduce. Cells that make up body tissues are held together by molecules like hyaluronic acid, a polysaccharide that forms a chemical glue. Some pathogens, such as certain Streptococcus species and Clostridium perfringens, secrete an enzyme called hyaluronidase that breaks apart this glue and makes space for the bacterium to penetrate deeper within host tissues. Collagen fibers, found at the base of superficial tissues, are another barrier to pathogens. Pathogens like Streptococcus and Clostridium secrete collagenase, an enzyme that helps them to pass through the barrier by digesting collagen fibers and allowing these pathogens to move deeper into the body. Streptococcus pyogenes, which causes strep throat and necrotizing fasciitis, secretes an enzyme called streptokinase that digests blood clots by breaking down the fibrin proteins that provide the structure of the clot. For this reason, streptokinase is sometimes called fibrinolysin. Some strains of Staphylococcus aureus secrete staphylokinases that do the same thing. Both of these species of bacteria live on the skin. The ability to dissolve blood clots allows them to penetrate deep beneath injured skin and cause a more serious infection. Enteric or intestinal pathogens possess a number of virulence factors. Two of these pathogens are Salmonella, the etiologic agent of salmonellosis, and typhoid, and Shigella, which causes shigellosis. Both bacteria are able to live within their host cells as parasites. Let's see how virulence factors help them to do so. Salmonella has flagella that help it to move in its environment. Proteins on these flagella inhibit phagocytes, as do other proteins in its capsule and cell wall. It also has fimbriae for adherence to the luminal surfaces of intestinal walls. It uses a specialized secretion system to inject proteins called invasins into the epithelial cell that cause rearrangement of the actin in its cytoskeleton. This rearrangement causes the membrane to ruffle, allowing the bacterium to be engulfed. Once salmonella is engulfed by the intestinal cells, it remains in the vesicle, where it triggers an inflammatory response causing electrolytes and fluids to leave the cell. This results in secretory diarrhea. Inflammation also causes leukocytes to migrate to the region. When the bacteria are released from the epithelial cells, they either enter the bloodstream directly or by moving into phagocytes where they can grow. Shigella, unlike Salmonella, lacks flagella and cannot attach to the intestinal epithelial cells. They can only enter by attaching to M cells, which are specialized antigen sampling cells of the intestine. Shigella also uses a secretion system to inject molecules that cause the M cell to take it inside. Once Shigella is inside the M cell, it passes through to the other side within a vesicle. The secretion system has also released proteins that induce inflammation, bringing phagocytes to the area. Shigella can survive within the phagocytes by escaping the phagosome. The inflammatory response breaks apart the junctions between epithelial cells, allowing Shigella to spread. Another virulence factor of Shigella allows it to polymerize the actin molecules within the epithelial cells into a tail-like structure that propels it from one cell to another. Exotoxins are bacterial proteins that are secreted by a living bacterium into its surroundings. In a host, these exotoxins circulate and cause damage to host cells in various ways. Exotoxins can cause severe damage or death at very low concentrations. Exotoxins can be classified in different ways. One method classifies the exotoxins by the area of the host that is infected. For instance, neurotoxins are toxins that affect the function of neurons. Tetanus and botulism are caused by species of Clostridium that secrete neurotoxins. Enterotoxins, like those secreted by the cholera bacterium, affect the lining of the intestinal tract. The term cytotoxin refers to an exotoxin that kills or seriously damages host cells in general. Corynebacterium diphtheriae is a bacterium that secretes a cytotoxin that inhibits protein synthesis in host cells. Exotoxins can also be classified by their structure and function. This classification scheme has three categories: AB toxins, toxins that disrupt membranes, and superantigens. Most exotoxins are proteins with two domains, A and B. The A domain of the exotoxin is an enzyme that has a particular function. The B domain of the exotoxin is the binding component. It binds to a specific receptor, usually a carbohydrate on the host cell. Once the B domain attaches to the host cell uses endocytosis to bring the exotoxin inside. Then, the B domain opens a pore in the endosome, allowing the A domain to escape into the cytosol, where it can inhibit protein synthesis or interfere with the host cytoskeleton. This usually results in death of the host cell. Membrane disrupting toxins exert their effect without entering the cytosol. They can form protein pores in the host plasma membrane. or they can disrupt the phospholipid portion of the membrane. In either case, the cell lyses. Superantigens are bacterial exotoxins that stimulate an excessive immune response. First, it causes a proliferation of T cells. Then it causes the T cells to release excessive amounts of cytokines, which can stimulate fever, inflammation, and shock. This very intense response could lead to death. The toxins secreted by Staphylococcus aureus in food poisoning and toxic shock syndrome is an example of a superantigen. Endotoxins are a part of the outer membrane of the gram-negative bacterial cell wall. The endotoxin is the lipid A portion of the lipopolysaccharide, or LPS, in the outer membrane. Endotoxins are only released from the bacterial cell when the cell dies or during cell division. When bacterial endotoxins are liberated, often during phagocytosis, they stimulate macrophages to produce cytokines called interleukin 1. Interleukin 1 is a protein messenger that travels to the hypothalamus in the brain, causing production of prostaglandins. Prostaglandins cause fever, an early sign of an infection. Endotoxins and interleukin 1 also help to stimulate phagocytosis, complement activation, and antibody production by B lymphocytes. When endotoxin is released in large amounts or within the bloodstream, it can have more serious and even deadly effects. Massive amounts of cytokines, particularly interleukin 1 and tumor necrosis factor, cause loss of fluid from capillaries and vasodilation, both of which cause a lowering of blood pressure, often to dangerous levels, leading to shock. These same cytokines also stimulate blood coagulation, which makes the shock even more severe. This deadly situation most often occurs in bacterial sepsis caused by E. coli or meningococci in the bloodstream or spinal fluid.